Today we are going to discuss about a practical class, that is inorganic qualitative analysis. Inorganic qualitative analysis, we are going to detect the ions in a given sample. Totally we are going to detect 4 ions, that is 2 cations and 2 anions. First, let us see about anions. The common anions are borate, fluoride, phosphate, oxalate, nitrate, chloride, sulfate, carbonate, etc. Here we are going to see about mainly 8 ions. So these 8 ions are further divided into two types, that is interfering and non-interfering radicals. So in a given sample, definitely there will be a interfering and non-interfering in an anions. We have to find two anions. So in that two anions, one will be interfering and other will be non-interfering. In interfering, we have borate, fluoride, phosphate and oxalate. In non-interfering, we have nitrate, chloride, sulfate and carbonate. Further, we are going to see the testing methods for interfering radicals and non-interfering radicals. Now we are going to see the test for anions. Already we have separated anions into interfering radicals and non-interfering radicals. Now in interfering radicals we can see borate, fluoride, phosphate and oxalate, and how to conform these radicals. First we can see borate. In borate, ethyl borate test. To conform borate, we must do ethyl borate test. The given substances is mixed with concentrated sulfuric acid and ethanol. This mixture is heated. While heating, the green edged flame is obtained in the edge of the test tube. This confirms that the borate is present. Next we can go to fluoride. In fluoride, the test with the concentrated H2SO4. The substances is mixed with concentrated sulfuric acid and heated. After heating, the sides of the test tube will appear as oily droplets. This is the confirmation of fluoride. On going to phosphate, ammonium molybdate test is done to confirm the presence of phosphate. The substances is mixed with concentrated nitric acid and it is heated and cooled. After cooling, ammonium molybdate is added. After adding ammonium molybdate, we get yellow precipitate, and this is to confirm phosphate. Oxalate test with dilute H2SO4 and KMNO4. The substances is mixed with dilute H2SO4, that is dilute sulfuric acid, and it is heated. After heating, we have to add a drop of KMNO4 solution. If the decolorization takes place, it confirms the presence of oxalate. If no decolorization takes place, it confirms the absence of oxalate. Now, non-interfering radicals. In non-interfering radicals, we are going to see nitrate, chloride, sulfate and carbonate. First, in nitrate, dry test is done. The substances is mixed with concentrated sulfuric acid and also we added copper tunings, and this mixture is heated to get reddish brown gas. The reddish brown gas confirms the presence of nitrate. Next, brown ring test is also done to confirm nitrate. The substances is mixed with dilute HNO3 and freshly prepared FeSO4 solution is added, and for that solution concentrated sulfuric acid is added dropwise. Here we get a brown ring. On going to chloride, chromic chloride test is done. The substances is mixed with concentrated sulfuric acid and K2Cr2O7 solution, and that mixture is heated to get a reddish brown gas. Here also reddish brown gas indicates the presence of chloride. Next we are going to sulfate. Barium chloride test, BaCl2. The substances is dissolved in dilute hydrochloric acid solution and it is heated and cooled. After cooling, the barium chloride solution is added to get white precipitate. The white precipitate confirms the presence of sulfate. Next, carbonate. Carbonate, the test with dilute hydrochloric acid. First, in a test tube, dilute hydrochloric acid is taken, and further the substances is added to check the brisk effervescence. The brisk effervescence confirms the presence of carbonate in a given sample.